So I just finished talking about protein functions. If you want to know more about protein functions, go in your textbook and read it in more detail. Now I'm going to continue with the amino acids. So in this slide, I'm going to talk about how proteins are built. And the first thing that I want to tell you is that, okay, we have been talking about that proteins are a biological molecule that is in charge of different functions, and they are formed. If you see this poly, they come from a basic unit building block that are the amino acids. And when you start getting together these amino acids through peptide bonds, you find peptides. And when you start adding more and more, you form a polypeptide. And then when you have, here I show you one polypeptide, but when you have two or three polypeptides, depending on the protein, they start interacting and forming a protein, and they start bending in different ways to have a three-dimensional structure. And the three-dimensional structure is very important to determine the function. So a protein is a biological function molecule that consists of one or more polypeptides. Proteins are linear polymers that are built from amino acids. Yeah, so you go from amino acid, form a peptide, the peptides start interacting between each other and they form a protein. Now I'm going to talk about amino acids and the structure of the amino acids. Amino acids are formed of four main components: the amino group, the carboxyl group, and remember that a carboxyl group is uh, an acid, yes, it's an organic acid. For that reason, amino acids are called amino acid, yeah. So because they have these two groups. The third component is the alpha carbon, and so far all these amino acids will have these three components. They will all look very similar. The difference between these all the amino acids is in the side change or R group. And why the R group? Because the R groups can be vastly different. Some of them can be very small. Some of them can act as a base. Some of them can be act as an acid. Some of them can be very big. So all the properties, chemical properties of the amino acids will come from the R group. Okay, so let's recap all the things that I said in this slide. I told you that the amino acids are organic molecules that are formed of a carboxyl and an amino group. So again, amino here, an acid from the organic acid that is the carboxyl. So amino acid. The second thing that I told you is that the following are some example of amino acids in the three main components. If you remember, the amino group, carbon group, and the alpha alpha carbon. So all of them look very similar. The difference come from, as I said before, that I want that you remember, come from the R group. So all of them look similar except for the side change or R group. So let me show you some R groups in a second. So amino acids differ in their properties due to the side change called R groups. And these are some examples of R groups. So for example, in glycine here is only one hydrogen, so it's a small R group. However, here you have, for example, tryptophan, you have two rings together. Well, here in the phenylalanine, you have one ring, and there is a lot of variation in all of them. And this is why they have such a different properties. We're going to talk about the type of amino acids. So They are some amino acids that are non-polar in the side change, so they are hydrophobic, like these ones. There are ones that are polar, and as they are polar, they're hydrophilic, like these ones. And in other cases, they can be also electrically charged. Their side change, like these ones. These two act like an acidic, negatively charged molecule, while this one will be basic, but they will be positively charged. In this slide, I want to show you how you can go from an amino acid to a protein. And to that, how you synthesize protein, you need to have this chemical reaction that releases water. But let me walk you again to all these schematics. The first thing that I want that you see here is that here you have two amino acids that are together. And why they are together? Because they have a peptide bond. While this one is separated, so you're going to add it. Here you have all of them are together. How? By peptide bonds. So you have three peptide bonds. And here you have the side change. They are groups. While this one are the backbones. Another important thing that I want that you remember, and I'm going to put it in a different color, is that always you're going to have on one side an amino end. So if you see here, there is an amino end. Here there is an amino end. Here there is an amino end. And now in green, I'm going to show you that there is always a carboxyl end. So here you have a carboxyl end. Here you have a carboxyl end. And here you will have a carboxyl end. And now let's see how you create the new peptide bond. So to do that, you have to release a molecule of water coming from the carboxyl end. So you have this carboxyl end, and you have to take the amino end, and these two form a molecule of water, and the water will be released. And in this way, you can create a peptide bond. You can create many different peptides that all of them will be different because of the sequence. So you can here we have this side change, but we can also have a different length, a different sequence of amino acids together. So to summarize, I told you that amino acids are linked and how they are linked by peptide bonds. So this is fundamental. I also told you that each polypeptide has a unique linear sequence of amino acids, but in all the cases, you are going to have a carboxyl end. So here you have a carboxyl end. Here you have a carboxyl end, amino end or N terminus. So here you have the amino end and the N terminus and amino end